Hello and welcome to the class on population mean. We know the basic model in quantitative genetics which describes phenotypic value as an expression of genotypic value plus environmental effects. The phenotypic mean of the population is based on the phenotypic value for all the individuals for a particular trait, and it can be calculated by taking sum of phenotypic value of all the individuals divided by total number of individuals in that particular population. In this presentation, we will try to understand the concept of population mean in terms of genotypic values and gene frequencies. Genotypic value. The genotypic value refers to the combined effect of all the genes influencing a particular trait. The genotypic value can be estimated by taking mean of a large population of the same genotype which is raised under similar environment. Now, the mean of this large population which is containing same genotype in uniform environmental conditions will assume that the environmental deviations are randomized and therefore the environmental effects are nullified and then the E becomes 0 in the basic model of quantitative genetics. We will try to clear the concept of genotypic value with the help of a hypothetical example. Suppose there is a locus with two alleles, dominant A and recessive A, which are affecting a particular trait. Then in the population, capital A will form a homozygous dominant genotype, capital A small a will form heterozygous genotype, and small a will form homozygous recessive genotype. Now, for calculation of genotypic value of homozygous dominant capital A genotype, we will consider an environment containing large number of individuals of a single genotype homozygous dominant capital A. Now, the phenotypic mean of all these individuals will represent genotypic mean of homozygous dominant genotype, because this genotype is measured in a large quantity under uniform environmental conditions. The genotypic value of heterozygous and homozygous recessive genotype can be estimated in the same uniform environmental conditions those provided to homozygous dominant genotype. Here, in this same uniform environmental conditions, we are measuring the phenotypic value of all the heterozygous individuals, and the mean of this phenotypic value will represent the genotypic mean of heterozygous genotype. Similarly, the genotypic value of homozygous recessive genotype will be calculated in the same environmental conditions which have been provided to homozygous dominant as well as heterozygous genotype, and therefore the phenotypic mean of all these individuals will then represent the genotypic mean of homozygous recessive genotype. Now, since the environmental effects have been randomized and E became 0, therefore the basic model P is equal to G plus E will become P is equal to G. Now suppose the genotypic values of homozygous dominant, heterozygous and homozygous recessive genotype are assumed to be plus A, small d and -A respectively, and the scale of genotypic values is expressed by taking 0 as the mid value of two homozygotes, that is homozygous dominant and homozygous recessive genotype. In this scale of genotypic value, small m represents the mean of two homozygotes, and here m is taken as point of origin, that is 0. This point of origin 0 denotes that plus A and minus A are being placed at equal distance from 0 or m. Now, the genotypic mean of this particular population can be calculated with the help of frequency of respective genotypes and their genotypic values. In this table, we can see that frequency of a particular genotype is multiplied with respective genotype value, and the genotypic mean denoted with G bar is calculated by taking sum of frequency multiplied by genotypic value divided by sum of genotypic frequencies, which upon solving yields a formula A into bracket P minus Q bracket complete plus twice PQD, which denotes genotypic mean of the given population. The genotypic mean represents deviation from the mid homozygote values, that is m. Now if we add mean of two homozygotes in the genotypic mean, it will yield population mean in absolute units, that is the actual units of the trait. So the population mean represented with m bar can be calculated with formula A into bracket P minus Q bracket complete plus twice PQD plus small m. Based on this talk, we have an exercise for you as your homework. This problem states that a trait is governed by two alleles, dominant B and recessive small b. The phenotypic values of various genotypes are measured under uniform environment. Based on the following information, calculate the gene frequencies small p and small q. Estimate the genotypic values small a, small d and minus a. The mean of two homozygotes, that is small m, and determine the population mean based on genotypic values and gene frequencies. Thank you.